Yeah, I just want to get your attention. Woo. So we're going to go ahead and get started. Thank you guys for all coming. Summertime is, I guess, tough, but we're glad to have you here. So I think do we have a couple announcements, slides, and stuff like that? Maybe not. We just usually go through our role. We got a lot of stuff going on here this summer. I've got pretty much all the Superheroes of Science booked for the next few months, and I'll get those out. September, Ed Kaloje has not agreed to it yet, but he's at Urban Waters, but I'm going to twist his arm. And you just tell everybody tonight, if you know him, that David said he's talking in September. He hasn't given me a clear answer. But I have an October speaker, a November speaker. So October is Chris Schell, who is a UWT professor also, and he does a lot of urban wildlife, and so a lot of wearable tech, camera-triggered technology. I have a November speaker, which I'm blanking on, but December is Martin Laderick, and he has a company called Bioscale down in the Bay Area. He's been connected with many companies, and I've known him, and he's a great person to have. So we're going to have him through. I also am working on a couple of other people for coming up, so we'll stay tuned. I'll get everything in. Okay. Am I looking at other slides? I guess I'm going to run through other stuff. So we have a number of programs here. We do Girls Who Code. So actually, can I see the raised hands, volunteers, Girls Who Code, people who are helping out with Girls Who Code? That's right. So talk to Christine if you want to volunteer, you want to bring kids. So it's pulling our 7th and 8th grade girls into this. We just finished a biotechnology summer camp, which was amazing. We had 15 students, and all our iGEM team kind of co-mentored that. So that was Jenna, and it was incredible. And so we're going to have our metrics coming out on that. We have, of course, the iGEM team. iGEMers, raise your hands. Woo! Okay. So we are getting ready. We have a competition coming up. We're going to go to MIT in November. We need all kinds of help for that. So if anybody's interested in helping us support that, that is going to be incredible. I could tell you about the project, but I'm letting them practice. So please go talk to them. If you need to know something about rhizobium or nitrogen fixation, that is the team to talk to. Okay? We have representation from our biotech companies here. I think Tinder's up in the lab. Stan, where are you? Stan's on there. And then you got Scott. These are 3 of our big biotech companies. I got somebody pointing over here. Tinder's right there. So these are people, if you want to talk about jobs in biotech or what they're doing, those are people to talk to. They'll take you up through the labs. And they're really going. So, okay. So I guess I am going to introduce Tom. So our Superheroes of Science. Oh, if anybody has any questions about what RAIN is or what they do, come talk to any of us afterwards, and we'll give you the brief on it. I'm also here to take tours of the lab. Oh, ground rules. 2, we will have questions afterwards. You please ask them. If it goes on longer, please feel free to leave if you need to leave. But if you want to stay and talk, meet a scientist, do things like that. Really come on up and interact. And we encourage lots of questions. Okay, so please do that. So I'd like to meet Tom Iliffe. I am so excited. He's been crashed at my house, and so we went diving. He's a professor at Texas A&M. He is a cave diver. If you saw any of his things, he was on PBS last night. He's been connected by Adam Savage, so if you know Mythbusters and all that. He's been on Nova, National Geographic. He's cave dives all over the world. I'll steal your thunder, but he has discovered species, 200 species of crustacean. Whole orders have been defined because of him. He's still holding out for his own phylum, and that's coming up. But he's really got a class of organisms. So really amazing. And so great thing, talking to him for the last few days, really understanding how you can take something you're passionate about and get funding for it and get to travel all over the world and do amazing research and then connect with other people and do research. So we're looking for ways we can collaborate with some of the technologies we have, and hopefully we've got his attention by bringing him up to Tacoma and interacting with the community here. So I want to give everybody just welcome to Tom, and I'm going to let him give the talk.
Thank you. It's a great pleasure to be here. I'd like to spend some time with you tonight. So we're going to take a trip. Everybody want to take a trip? Okay. We're going to go on over to the dark side. Okay. We're going to go down into pitch black underwater caves. We're going to go a long way in and a long way down. And we're going to see what the environment looks like first, and then we're going to see who lives there. So what's going on there? How does the systems work? When I started doing this, quite a few years ago, there was people told me there isn't anything there. It's a lifeless environment. Forget about it. I didn't quite believe them. So I've been exploring, going to places where no human has ever been, doing things that nobody else has ever done, and making discoveries that are totally unknown to science. So we're going to see all sorts of things going on tonight. The first half of my talk, we're going to look at places. So we're going to try to understand places. We're going to see what's going on there, and then we're going to see who lives in these places. So part one, places. Part two, who lives in places. So I've been fortunate to have my work featured in a number of venues. We've had cover stories on National Geographic magazine. We've been on a number of diving magazines. We've been on covers of a number of scientific journals. So we've been doing very well with publications. So I've published to date more than 250 journal articles and book chapters. So we work very hard to get information out into the scientific literature. Okay, so the caves that we work on are called Anchialine Caves. So the word Anchialine comes from the Greek, Anchialos. It literally means near the sea. So very simply, these are caves near the sea. That's the definition. Pretty simple. So they're caves that contain tidal saltwater pools. They're pools that have no surface connection with the ocean. Their connection with the sea is totally subterranean. So this is an example of a saltwater pool, an Anchialine pool, a cave pool near the sea. And if you look at the back wall of the pool there, you can see the tide line. So this photo was taken at low tide, and the tide goes up and down. There's fluctuating. As the tide goes up and down, water is being pumped in and out. So it's a dynamic system. It's a system where things are always happening, parts are always moving around. And diving is a critical component to the work we do. The reason is that these caves contain a layer of fresh to brackish water on the surface. Brackish is a mixture of salt and fresh water. So not pure fresh water, not pure salt water, but half and half or something like that. And then at some depth in them, they have a halocline. A halocline is a boundary between this lower saline water and the fully saline ocean water down at depth. And most of the animals we find are in this deep ocean water. The only way to get down to the deep ocean water is by diving. Diving is an absolutely essential part of all the work that we do, and it was when we began diving in these cave systems that we started making all of our discoveries. Now, tools. We have a toolbox, and one of our tools, instead of using tanks, scuba tanks on our back like most cave divers, we use what is called side mount diving. So the tanks are under our arms, and because we need redundancy in what we do, we need a backup plan in case something goes wrong. What are we going to do next? We need a plan B, okay? Our plan B is to carry extra of everything. We carry 2 tanks instead of just 1 scuba tank. And we carry one of them under each arm. Under each arm, there we can look at the tank valves. We can check and make sure that everything's good with the valves. We can see if there's a problem. We can deal with it very easily. It's right in front of us rather than on our back hidden away where we can't see what's going on. So this is our preferred method of diving, is using this side mount technique. Now, normally we take at least 2 tanks, but we can take 3 tanks, we can take 4 tanks, and we clip them on the same way in the same position. So we can clip on multiple tanks, and we can drop these tanks off if we need to. So there's a very simple clip. We can unclip it, and we can leave it at some point on the line. So typically on a dive, we might take 1 tank, breathe 1/3 of it, and then clip it to the line, and then switch over to our double tanks and begin the dive from there. On the way back out, we pick up the tank we left and breathe from it on the way out of the cave. So we always want to have extra. We carry 3 lights. We use a line, a guideline, to find our way around through these caves. They're pitch black, no way to navigate, so we have a thin braided nylon line that we lay tied on outside the cave, and we follow that all the way through on our dive. We always have a way to find our way home. That's the most important thing, is not to find your way out, or not to find your way in, but to find your way back out again. You want to survive your dive. Okay, I've been doing it for a while. I'm still alive, so I guess I've been successful at what I do. We also recently have been switching over to a closed-circuit rebreather. And this is made by a company very close by, down in Centralia. So just this morning we were down there and I was visiting with the owner of the company who invented and developed the product that we're using. This is called a Megalodon closed-circuit rebreather. Closed-circuit rebreather is very different from normal scuba. In normal scuba, every time you take a breath and exhale, all your exhaled gas is lost as bubbles. And then you take a new breath, it comes out of your tanks, you exhale, and it's lost to the water. With a rebreather, every time you inhale, you get gas from the system, from breathing bags that are on your chest, and when you exhale, your exhaled gas goes through a carbon dioxide scrubber, removes all the CO2, you add a little bit of oxygen to compensate for the oxygen that your body has used, your cells have used, and you rebreathe that over and over again. So no bubbles, and all of your oxygen is going into your tissues of your body. All of your carbon dioxide is picked up and contained by the unit. So with a unit like this, I've done dives as much as 6 hours long. So I can stay down a long, long time underwater, and my decompression, the fact that on the way back up I have to stop and let nitrogen out of my system, is much improved, and decompression times greatly reduced. So I'm breathing the optimal gas mixture at all times during the dive. If you're familiar with nitrox, you're only breathing the optimal mixture at the deepest point of the dive, at one point and one time. With this system, I'm breathing the best mixture at all times during the dive. Okay, so there's a lot of advantages. However, there's a few small, minor disadvantages. On the inside of many of these units is a little sticker, and it says that this unit is trying to kill you. Your job is not to let it succeed. So it takes a lot of attention. Every 2 to 3 minutes, you have to be checking your oxygen levels in your unit. If your oxygen levels get too high, you go into convulsions and you die. If your oxygen level gets too low, you pass out and you die. So other than that, there's not much problem, right? So it takes a lot of training, and especially using one of these in a cave. In the ocean, you can always go straight up if there's a problem. In a cave, you've got a rock ceiling over your head. You can't just go straight up. You have to go back out the way you came in. Okay, no other way about it. And so the bottom line is, in a cave, you're a long way from home. I've been in caves as much as nearly a mile in. Okay? Think of that in terms of the ocean. To get an equivalent distance from the surface, you'd have to be a mile deep underwater. Okay? A long way from home. So you need a lot of special care, special attention. You need to make sure that your unit is functioning properly, and you have to have a plan B at all times. Always have a backup system. Okay, so where do we go? Where do we use this? So here's a map of the Atlantic Ocean. But normally when you see the Atlantic Ocean, you see this flat blue stretch of water. Well, really the Atlantic Ocean isn't like that at all. Here's the Atlantic Ocean if we took all the water out. If we remove the water, there's a big, huge mountain range running right down the middle of the ocean. This is called the Mid-Atlantic Ridge. And actually, the continents are moving, so they're separating. Europe and North America, South America, Africa are drifting apart. This is a process called continental drift, okay? So the youngest rocks in the Atlantic Ocean are located right down that ridge in the middle of the ocean. The oldest rocks are located closer to the continents. The closer you get to the continent going either direction, the older the rocks on the seafloor. In the middle of the ocean is the youngest rocks. So we're continually pushing up new seafloor and pushing the continents further and further away from each other. So where do we go for our studies? So we're working in the Atlantic Ocean here, and we do work in the Bahama Islands. So the Bahama Islands located just off the coast of Florida and north side of Cuba. So east side of Florida, north side of Cuba, Bahama Islands. Work in the Yucatan Peninsula. Yucatan Peninsula is part of Mexico. It's this peninsula of land that juts out into the Gulf of Mexico and separates the Caribbean Sea from the Gulf of Mexico. Another place we work in the Atlantic is the island of Bermuda. Bermuda is a volcanic island, was formed by eruptions about 35 million years ago, and is about 600 miles off the east coast of the U.S. 600 miles from any land. So it's well out to sea. This is where the Bermuda Triangle stories came about. I lived in Bermuda for 11 years, so I got to know Bermuda and Bermuda's caves very well. So now we're going to flip over to the opposite side of the Atlantic Ocean, and off the coast of Morocco, off the coast of the Sahara Desert in North Africa, we have the Canary Islands. And we're going to look at caves in each of those locations. We're going to compare and contrast how these different caves and different environments and different cave animals are from one to the other. So, first stop is Bermuda. So Bermuda is a volcanic island, but the top of this volcano has been capped over by limestone. And the limestone comes from coral reefs. So because the Gulf Stream carrying warm water out into the Atlantic, Bermuda has warm water, has the northernmost coral reefs in the Atlantic Ocean. And these coral reefs have formed a ring completely around the island of Bermuda. Bermuda, as Mark Twain said, it's shaped like a fish hook, only smaller. Okay, so here we have the fish hook island of Bermuda, and Bermuda is where I began these cave studies. It's all limestone. All of the island itself is made up of limestone. There's nowhere in Bermuda you can actually see the volcanic rock. It's all deeper down. So there's a number of caves in Bermuda. They're limestone caves, and we began doing some diving explorations in these caves. So these caves are quite deep, quite long. You can see our guideline there that we've laid through the cave to find our way around, to navigate. And these are permanent guidelines that we leave in place in the cave, so they're always there. And if we want to explore a new area, we set up a new line that we run out from the older existing lines. One of the things we noticed right away is that there are massive underwater stalactites and stalagmites in these caves. Now, stalactites and stalagmites are what you see in normal limestone caves. They're formed by dripping water. So dripping water can only occur in air. You can't have water dripping underwater. It doesn't work. So these caves must have been dry. The only way we could get stalactites and stalagmites is to have a dry cave to begin with. And the only way these caves could have been dry is that sea level must have been much, much lower. This was during the ice ages. So during the ice ages, sea level was as much as 300 plus feet lower than it is today. So 300 feet down, all of the caves in Bermuda that we know of would have been dry and air-filled caves. And so these are dry caves that were now submerged by rising sea level at the end of ice ages that were able to swim around and venture into for the first time ever. We're the first humans ever to go into these caves. We see things that no human has ever seen before. Where do you go to see things? You know, climb a mountain. People have been there before. You can fly over a mountain in a plane. You can see what the top of the mountain looks like. When you get to the top of the mountain, you know you're there. But in a cave, you have no idea. It's a mystery. And there's always a chance that you miss some little hole going off and that the cave goes much further, much deeper, much more is there. So you're never done. There's always discoveries to be made. Okay, so time is put on hold in these caves. It's like we're going back into a cave that is just like it was at the end of the ice ages. So as soon as water levels moved up and submerged these stalactites, they stopped growing. So they've been in this way, in this form, for at least the last 10,000 years. So nothing has happened in 10,000 years. When we go in here, it's seeing what the cave looked like 10,000 years or more ago. It's cool. I love it. Now, okay, so the question is, we have animals in these caves. The caves were all dry and air-filled. So where did the animals that we find today in the water-filled caves, where did they live when the caves were dry? The animals didn't come into the caves after the caves flooded. The animals are very old animals, exceedingly old. So they had to have a place to live when sea level was lower. So we started a project to look for deep caves, and we went to the edge of the island, and we started diving off the edge of the island. At a depth of 200 feet, we found a cave here. There was a tunnel that extended back. You can see that's me in the background there, and behind me is some blue water of the ocean. Very, very clear water. You can see ripples of sand on the floor of the cave. Unfortunately, this particular cave didn't go very far. So it was a tunnel that is collapsed in, probably at one time was part of a flow system, but now has collapsed in. But it's still too shallow because 200 feet down, it still would have been a dry cave during the ice ages. We need something even deeper down. So we got to go deeper. So with our trusty rebreathers, we went down to a depth of about 370 feet. And at that depth, we found a ledge that was cut into the wall, into the side of the island of Bermuda. And this was a place where waves pounded against the cliff. And as they pounded against the cliff, they cut a notch into the wall of the island. So this is the place where the lowest point sea level reached during the ice ages. 18,000 years ago, there were waves pounding against that cliff at that point. We've gone back in time now 18,000 years. We got our time machine ramped up. We're moving. So, not deep enough. This was still all dry. Not deep enough, not good enough. We need to go deeper. We need to go deeper. So, we went down. Our deepest dive was to a depth of around 400, close to 450 feet. At this depth, we're well below where sea level was during the ice ages. And at this depth, the water is still clear enough, there's enough light coming through, that we don't need any artificial lights. It's really amazing diving. So, very beautiful diving. Actually, for these dives, we were out on an offshore seamount 20 miles away from the island of Bermuda. And we're diving on the edge of this seamount. For our dives, we have a maximum of 15 minutes from the time we leave the surface, do all our descent to 450 feet, take a quick look around, and when our watch says 15 minutes has gone by, it's time to ascend. It takes us about 2.5 hours to do our descent. So 15 minutes to go down, 2.5 hours to come back up. Unfortunately, because of this limited time, we didn't have much chance to look around to see what's there. But we have an idea now that there is a sheer cliff, and we could just look down from this cliff, and it was going down, down, down. We could see a long way below us. The water is very, very clear. There's a lot of light down there, even at that amazing depth, and a lot of life. We can see all sorts of encrusting forms growing on the cliff face here. Okay, on the way back up, we need some friends to keep us company. So we call in a giant manta ray, swim around with us, keep us company, and move our way to the surface. Next, we're going to go to another island group, the Bahamas. So the Bahamas, we said, is on the east side of Florida, and it's a chain of islands here. So this is what the Bahamas looks like from space. There is a series of islands, but these islands are located on a shallow water platform. So here's this shallow water platform. This is called the Great Bahama Bank. If you look up to the top, there is another platform called the Little Bahama Bank. But separating these two platforms is a channel that's about 1 mile deep water. So there's isolation between the platforms. We have caves on both sides of this channel, and we're looked at biology, what's living in caves on opposite sides of this channel. So we have a lot of isolation going on, and isolation, as Darwin would say, is one of the key components of evolution. You need to, when he went to the Galapagos, he found that on different islands in the Galapagos, the birds, the finches, form different species on each island. Here we have caves that are on different oceanic islands, separated by deep water channels. We want to see what's going on with the animals in this environment. So we're doing a underground Darwin experiment. I wish he was here to go with me. So the caves in the Bahamas are called blue holes. So this is the local name, and they're blue because the water is so clear, very blue water, very deep holes, blue holes. And there's two types of blue holes. There's blue holes in the ocean called oceanic blue holes, and on top we have an ocean blue hole. It's connected around by a bay that comes in. Here you can see half a dozen people or so standing near the edge of it, so you can get some idea of scale. This is called Dean's Blue Hole on Long Island in the Bahamas, and the depth here of this blue hole is 660 feet deep. So vertical shaft going down 660 feet. Amazing depth on these systems. Inland blue holes are out in the interior of islands, and they're circular, very circular formations, very clear water, and oftentimes quite deep. This one is a little more than 200 feet deep. Let me run through this very quickly. There's 4 parts to this slide. So the top left corner here is salinity, so we're measuring the salt content. And we have depth, water depth on the vertical axis and salt content on the horizontal axis. So we start out at the water surface and we have low salinity, low salt content in the water. If we go down to a depth of about 60 feet deep, there is this boundary between salt and fresh. And all of a sudden, very quickly, it goes from fresh water almost to pure salt water. And then once you're through that, all the way down, it's all salt water. So we have 2 types of water. Basically, fresh water on the top, salt water on the bottom. Okay? Very simple. But when we go to other parameters, it gets much more complicated. This is in the same cave looking at water temperature with depth. So increasing temperature going from left to right, increasing depth going from top to bottom. And we start out with temperature dropping, getting colder, and then as we get this halocline, the boundary between fresh and salt, all of a sudden it warms up, gets really warm. And then once you get through that into the salt water, the temperature drops down again. So there's a lot of fluctuation going on in these systems as far as temperature. Here is pH or acidity, and we start out with relatively higher pH. The pH drops to a low point right at the halocline, and then goes back up as we go below the halocline, and then starts dropping back down as we go deeper and deeper. So it's bouncing around quite a bit. If we look at dissolved oxygen, how much oxygen is there for organisms to be able to respire? Surface water is in contact with the atmosphere, where there's a lot of oxygen, and so we start off with a lot of oxygen in the water. We drop down to very low to even almost no oxygen just above the halocline, the salt water. As we go into the salt water, all of a sudden there's a lot more oxygen available. So here's a good place for animals to live. There's oxygen for them to utilize. And then as we go deeper and deeper, the oxygen disappears and there's no oxygen at all. So a lot of interesting fluctuations going on in the water, and all of this is related to the biology. Okay, we're going to move on to the Yucatan Peninsula in Mexico, and the caves there are called cenotes, very much like the caves in the Bahamas, just a different name. So instead of calling them blue holes, we're going to call them cenotes. Cenote is a Mayan word for natural wells. So these are natural wells. In the Yucatan Peninsula, no surface rivers or streams. All of the water transport is underground, all subterranean. So for the early ancient Mayan people, the only way to get water, drinking water, was to get it out of these natural wells. This is underwater what they look like. Pretty damn spectacular. It's really going into these things and seeing something like this, you know, literally, wow, takes your breath away. You don't see anything like this anywhere else. Just this laser beam of sunlight coming down into the black. And most of the time they're spreading out, they're getting larger and larger and larger the deeper you go down. So the smallest, narrowest part of these systems is right at the surface at the entrance. Gets bigger and bigger and bigger the further you go. Some caves have some very unusual, unique types of formations. This is called Hell's Bells. It's only found in one cenote in the entire Yucatan Peninsula. It's at a depth of about 140 feet down, and it's just into the salt water. It's really no good explanation as to how these have formed. And if you see behind the diver there, there's a lot of them. It's not just one or two, there's a lot of them. And they're very big. They're huge. So a lot of questions on what's going on in the environment, how the environment, the changing environment, whether it's microorganisms or whether it's changing conditions as far as acidity or oxygen or temperature or who knows what is going on. Now, the longest underwater cave in the world is in the Yucatan Peninsula. And this particular cave is located along the Caribbean coastline. It's Sistema Sakaktoon, and here's the Caribbean Sea, the blue area, and there's a whole series of network of tunnels. All of these tunnels are underwater. You need to be a cave diver to explore any of this. The system is 350 kilometers long, so 200 odd miles of underwater cave passages. Would take a long time to discover, to explore, to map 200 miles of underwater cave, and they're still finding more. The project isn't done. There's still a lot to do. So this is an enormous system, 219 miles long. Really, some incredible stuff happened in Yucatan. One of the most significant for us today, every one of you, occurred 65 million years ago. 65 million years ago, a giant asteroid came in and struck the planet right at the northern boundary of the Yucatan Peninsula. It threw up so much dust and debris into the atmosphere that this dust and debris orbited the Earth, came back in, and set off fires all the way around the planet. The whole planet was burning, not just the place where this meteorite struck. It was 5 miles across. It's huge, but it threw up all of the material where it hit the Earth and was orbiting the planet. There was a dust storm, total darkness, no light for months at a time. All of the plants died, all of the plant-eating dinosaurs died, and all of the meat-eating dinosaurs had nothing to feed on. They died as well. That was the end of the dinosaurs. And the chapter that ended the dinosaurs allowed for the evolution of mammals. So without that event, without that asteroid striking the Earth, mammals would be little mouse-like rodents running around under your feet. And I'd be talking to a group of dinosaurs in the room right now. So you'd be dinosaurs. Okay? If you want to be a person, this had to have happened. And it was a one-off event. How did this happen? Why did it happen? And how did it impact the entire planet and evolution of life on the planet? Okay, so when it hit, it gouged out a large part of the Yucatan Peninsula. This is a subsurface map of the meteorite crater, what it looked like. And around the edge of the crater here, the crater was covered with limestone over 65 million, intervening 65 million years. About 1,000 feet worth of limestone. And so we can't see the crater, but we can see a sign that the crater is there. So the white dots are all the cenotes, the natural wells in the peninsula, the caves. And there's a semicircle of cenotes that falls directly on top of the edge of the ring of cenotes. So I've done some diving, and I've been diving in these cenotes that are part of the ring. And so these cenotes are, diving in here is like diving into the barrel of the gun that killed the dinosaurs. Not many people can say that. I went down into the gun that killed the dinosaur. Man, pew, bam, got him. So a lot of very unique things going on. So we're going to move across the Atlantic Ocean, and we're going to move to a totally different type of cave. These caves are volcanic caves. Instead of being limestone formed by sediments, they're formed by erupting volcanoes. So we have a volcano that erupts, we have lava that flows down the side of the erupting volcano. The lava then solidifies as it cools down on the surface, continues to flow underneath. Eventually, the volcano stops erupting and the lava runs out the end, and we have a hollow cave-like structure, but in volcanic rock. So instead of taking tens or hundreds of thousands of years to form like a limestone cave, these volcanic caves can literally form almost overnight. Very quick. And these caves are formed at the same time the surrounding rock is formed. So the cave we're working on in the Canary Islands is on an island called Lanzarote. The cave starts at the base of a volcano and extends 3.5 miles down the side of the island as a dry cave till it reaches the coastline. At the coastline, there is a saltwater pool where we enter the water, and then we can go a mile out to sea in an underwater seafloor flooded cave under the Atlantic Ocean. This is called the Atlantida Tunnel, literally the tunnel to Atlantis. Okay? So we went from the gun that killed the dinosaurs to diving in a tunnel to Atlantis. I think that's kind of cool stuff. I don't know. And so this is the area of the pool where we go into the water, and this is what the tunnel looks like going out to sea. It's a huge tunnel. So it's about 60 to 80 feet high in places and 30 to 40 feet wide. The water is crystal clear in the cave, and it's just an amazing dive. We're using our closed-circuit rebreathers here because we've got a long swim, a mile-long swim to get to the end and turn around, and a mile swim to get back out. So 2-mile round trip. We're going to switch over to the other side of the world, see what's there. We went over to Christmas Island. Christmas Island is in the Indian Ocean now, and here's the world map here. This is the location of Christmas Island. It actually belongs to and is part of Australia, so it's an Australian province here. Now, if you go to the southern side of the island here, although you're basically in the tropics, and you look off the southern shore of the island and look straight south, the next landmass is Antarctica. So a long way to go in places. You're a long way from home. So these are all limestone caves. We're all diving in a rainforest on Christmas Island, a magnificent rainforest, magnificent trees here in the jungle, really beautiful, unique place. About half of the island is a national park, Australian National Park. As we go around the island, we find in the interior of the island big cave openings that we enter and go down into, and we go down far enough and we find these tidal saltwater lakes, the same type of lakes we've been finding everywhere else. So same caves, same type of caves, different side of the planet. So what's living in these caves? So first off, we'll return to Bermuda. So most of the animals in these caves are crustaceans, so very small relatives of crabs and shrimp, but oftentimes a much greater variety than we find on the surface. So we have an animal called an amphipod. We have a shrimp, in this case, bright red in color. Oh, the amphipod, by the way, no pigment, no coloration, and no eyes, although the shrimp does have eyes and does have pigment. This is another smaller shrimp. This particular shrimp was named after me. It's called Tiflatia iliffe. Here is one of the unusual animals that we found early on. That's a new order of crustaceans, so a totally higher group of animals previously unknown from anywhere on Earth. Moving over to the Bahamas, we have shrimp again, very similar shrimp to the shrimp we found in Bermuda caves. We have an animal called a thermospinacean, hot water shrimp literally, and these are animals that were originally described from hot water springs in Italy. And here they show up in caves on the opposite side of the Atlantic Ocean. Here's a small mysid shrimp, big eyes, but we can see its internal organs. And this is a very strange animal that we're going to hear more about in a few minutes, called a remipede. Remipedes represented a new class of crustaceans. So very much higher group of animals only known from saltwater caves and nowhere else on the planet. Yucatan. A lot of the animals, again, no pigment. Here we have a type of thermospinacean, hot water shrimp, but it doesn't live in hot water. That's just because the first place it was found was in hot water. We have an amphipod. Here we have an animal, looks like a little bivalve mollusk, but it isn't. It's actually a crustacean. It has jointed legs. Mollusks don't have legs. These guys do. Therefore, it is a crustacean. But it does have a double shell that the animal lives in. This animal up there is called a Stygiomycid, and this is a very unusual type of animal. Stygio referring to cave, so cave mycid. Christmas Island, opposite side of the world, we find these same type of ostracods in the caves, the bivalve shell crustacean. We have amphipods. We have some small shrimp, and here we found some unusual worms in the caves. So we got a lot of cool stuff. So we need to try to look at them in more detail, make some sense of them. Let's look at these ostracods, these bivalve crustacean animals. Now there's 3 different types that we found in general in the caves. So the first type is called Humphreyiella. They're named after a friend of mine, Bill Humphreys, a biologist from Western Australia, and there are currently 12 species. Of these 12 species, all 12 are only in saltwater caves, 4 of them from the Bahamas, 2 from the Canary Islands, 1 each from Cuba, Jamaica, Yucatan, Galapagos in the Pacific, Western Australia, and Christmas Island. So, opposite sides of the planet, but only in saltwater caves. Another type called Devia lacks these 3 spines that Humphreyiella has. They're pretty smooth all the way around. There's 7 species of Devia. They're only known from saltwater caves in the Bahamas, nowhere else. And finally, Speleacea. They have this little notch in the carapace, very characteristic, easy to distinguish. And there's 8 species, 4 from the Bahamas, and 1 each from Cuba, Jamaica, Yucatan, Bermuda. So all from basically the greater Caribbean area. So we have Humphreyiella all around the world, we have Speleacea, greater Caribbean, and we have Devia, only Bahamas. So some very characteristic distributions of those 3 different groups. Obviously, they're moving around into caves using different routes and are limited to different areas of the world. Now, Remipeds are what I told you about. We're going to spend some more time on these. So they're an enigmatic group of free-swimming crustaceans. They look like a centipede, but they're not. They're a crustacean. They have a short head, long segmented body or trunk, and paddle-shaped limbs. So they're continually swimming in the water. They never stop swimming. They're always up in the water column, never on the bottom. They're swimming around everywhere. So as we began working with these groups, we found more and more and more remipeds. Right now we're up to 29 species of remipeds. It's a new class, very important. We need to give very good names to these animals. Okay, it's a very serious thing because once we named it, that name's going to stick forever. So you really got to give a good name. These are very special animals. We need a very special name. So we thought and thought and thought. These animals are predators. They're vicious animals. They're top of the underwater food chain in the caves. So we need something for a big, they're relatively large for caves, vicious animal, something that would be really spectacular. So we thought and thought. Finally, it came to us. Of course, there's only one choice. Japanese movie monsters! Okay, so we've got the Godzilla remipedes. Everybody knows Godzilla, of course. We've got Mothra, the giant moth that was a foe of Godzilla. You remember those epic battles between Godzilla and Mothra? Oh, pretty cool. We've got Anguirus, the giant lizard. We've got Kumonga, the giant crab. We were kind of running low on Japanese movie monsters, so we switched gears and we went over to the Morlocks. Who can tell me what a Morlock is? Oh, what's a Morlock? Okay, H.G. Wells, The Time Machine. So the Morlock are the creatures that lived underground, and they went up and grabbed off the very beautiful young women and took them down into the caves. So we have all of these represented now in the remipedes. So we put together a phylogeny, family tree, so to speak, of the remipedes. Up on top, we have the Godzilla remipedes coming out up here. Then we have the Mothra, giant moth remipedes here. Over here are the Morlock remipedes. Down here, the Anguirus remipedes, giant lizard, and finally the giant crab remipedes down at the bottom. So they all separate out very nicely based on their position. We've got, of course, Godzilla has to be at the top, he's the king. Godzilla's the king. So this is how everything falls out, and as we put our science together. Okay, one of the things we looked at in the remipedes is we never found how remipedes were actually reproducing. And it wasn't until just a few years ago that we found larval remipedes for the very first time. So there's 29 species of remipedes. Right now, we know larvae from only one species in one cave. And this is what the larvae look like. And so when we first saw them, we called them backstrokers because they swim on their back, stroking and swimming with their antennae. So remipedes always swim upside down. The backstrokers always swim upside down. Even larvae have the same orientation as the adults do in their swimming. And we found a number of growth forms, a number of developmental stages of these larvae, starting at the top. Left corner is the youngest form, and going down and developing, and you can see they're elongating. They have their own nutrient food supply within the body, so they're not feeding larvae, they're non-feeding larvae, and as they reach a certain stage, then they switch over and look very similar to just a miniature adult form. So this is the development of the larvae. and why we only have one species represented. We know what the larvae look like, but we can't find them anywhere else other than one cave and one species. Okay, so a question is where do the remipedes fit into the overall family tree of the arthropods? Arthropods are the jointed leg animals, so these are the animals that include the crustaceans, include the insects, and include the spiders and things. So when we plug this into doing DNA sequencing and fitting it all together, up near the top is the remipedes. The remipedes turn out to be the closest related sister species to the insects. So if you want to look at evolution of insects, you need to learn how to become a cave diver and dive in saltwater caves. Why didn't people figure that out before? It's intuitive, right? Here's what the remipedes look like in a little bit greater detail. Very long trunk, paddle-shaped appendages, swimming through the water very gracefully. The material that we see is actually the material in their gut. We're seeing the gut contents in these animals. The remipedes are actually the only venomous crustacean. They have venom-injecting fangs, so they're able to inject venom through their fangs into smaller crustaceans or other prey, and then literally suck the juices out of the prey. So this is what the front part of a remipede looks like. You can see the two appendages up there. The brown fangs are sticking out from either side. So here's where they're grabbing onto you and sucking in. So if you want to create a movie monster, wow, you got a fantastic start with just looking at the remipedes. Another remipede here. This is one of the Godzilla's remipedes. Okay, here's Godzilla, and here again, look at these long fangs that grab onto and stick into the prey, and they've got hydrolytic enzymes that they inject into the prey to break the prey down. And Cryptocoronides, another remipede, very unusual pads on the appendages here, the mouth parts, and small venom-injecting fangs. Okay, so these are all of the animals that we see. We're going to see a little movie, and we're going to go diving into a cave. So what's it like to dive in a cave? What's involved in one of these expeditions? And we're going to have a little short video that condenses all of this experience together. Okay? So, can you?
Throw that in there.
So we want to do the Bandito. Oops, we don't have a picture. No picture. Can we restart it? Okay. Can you back it up?
Yep.
Start it over again. This is in Yucatan Peninsula in Mexico.
7 caves that we've been, there have been more people on the surface of the moon than have been in these caves. I'm Tom Island. I'm a professor of marine biology. My research concerns studies of life in underwater caves.
My name is Guillermo de Anda. I am an underwater archaeologist.
The caves that are found in the Yucatan Peninsula are called cenotes.
The cenotes had a very strong link and importance with the Maya civilization.
There's more than 5,000 cenotes known. 1 or 2% of these have been explored so far by divers.
Remember that moment.
Okay. Tom, what your little friends that you said that everyone told you weren't there? What's the scale? Are these 4 feet long, an inch long, microscopic?
So the remipedes are up to about 2 inches long or so, okay? The others are maybe anywhere from an inch long to quarter of an inch to even a bit smaller. So the water's so clear, we've got very bright lights, we scan around. If we see a speck in the water in front of us, it's an animal. There's nothing other than animals in there. So we collect animals in individual tubes. We have the tube, a water-filled tube, we take down, we unscrew the cap, put our finger in, pump the water out, and then collect each animal in an individual container and bring it up to the surface in the water where it lives, okay? And once we get it to the surface, we can look at it while it's still alive. We can take photographs of the live animals fresh from the cave. We can see what the color looks like, how they're behaving, and then we preserve in various ways. So we preserve in ethanol, or we preserve in a number of other special fixatives if we're doing sequencing work. So it's our lab is fully set up. It may be in our hotel room, okay? So we are a moving feast as far as our science is concerned. We bring our science and our lab along with us, and we do work, prepare specimens that we can then work with other collaborating scientists. I've got some projects going on right now looking on the brain and the nervous system of remipedes with scientists in Germany. So oftentimes they'll come over, join us on a trip, do special dissections, and then they'll take the specimens back home with them for future analysis going into much greater detail. So it's cool, but the temperatures here are ice cold, so cool to ice cold. So I'm a warm water diver, I'm spoiled. Virtually every place where these caves are is in the tropics. So we dive in tropical caves. We really don't have any of these areas, these cave animals, this environment, anywhere other than the tropics. But it's the circumtropics, it's the tropics around the planet. Okay, so temperature, normally we're wearing a wetsuit. We're very comfortable for long dives, and we're actively swimming the whole time, so we're generating heat that way. But it isn't like we don't have temperatures here. So typically 72 to 75 degrees Fahrenheit.
I got a question here. I'm going to ask one. So I have a question about, you're talking about your apex predators, and you alluded that they're eating other crustaceans. What is their ultimate food energy source? Okay. They're not getting it from the sun, are they?
No. So obviously there's no light, so no photosynthesis going on, no photosynthetic production of carbon. So everybody on the surface, doesn't matter what you eat, you're going to get it from either eating plants, or you're going to get it from eating animals that ate plants. It all comes down to plants, but no light, no photosynthesis, no plants means two things. One is no photosynthetic carbon, no food, and also another thing is oxygen. The oxygen in our atmosphere is produced by plants. No plants, no oxygen generation. So we lose two things in the underwater caves. We don't have the production of organic matter, of food, and we don't have the production of oxygen to breathe. So you're going to starve to death and you're going to choke to death. So the animals have to work a way around that. We believe that we found evidence that there is chemosynthesis, organisms that are able to utilize chemical energy rather than light energy to produce organic matter. And so what is the chemical energy? Well, one of my PhD students just finished a project looking at methane generated organic matter in underwater caves. So it's there's methanogenesis and breakdown of methane that is a way for these cave organisms to generate organic matter from bacteria. So bacteria are utilizing the chemicals in the water, and then the animals are feeding on the bacteria or are feeding on other animals that fed on the bacteria. We're still working all of this out, but the pieces are starting to fall into place.
That was my question about the composition of the water that's so clear. Are there chemicals and also minerals that really the animals feed on or utilize?
It's salt water. Just
I'm not used to these things.
same as in the ocean. So it's salt water. You have, it's, for the most part, in the limestone caves, there's a lot of dissolved carbonates in the water. Limestone is calcium carbonate.
Why don't these stalagmites and stalactites break down from limestone so?
Because the water is saturated with calcium carbonate. It can't dissolve anymore. So it's already dissolved as much carbonates as it can. It can't dissolve more. And so these formations have remained in place for many tens of thousands of years.
That all leads into one of my questions. Is thermocline, chemocline areas of the cave, is it a multi-level chemocline?
Sometimes there's multi-levels of haloclines, chemoclines, and typically there's either one or two. So in one of the caves we're working at about 15-foot depth is the first. It goes from freshwater to brackish water. Brackish water is about 50% fresh, 50% salt water. And then at a depth of about 60 feet is a second halocline, and it goes from the 50-50 salt concentration to 100% salt, or meaning 100% relative to the ocean anyway. So same salt concentration as the ocean. That's, well, ironically, the methane is being generated at the surface in the soil, being carried down by rainwater into the upper parts of the cave ecosystem. So it's at the shallower places we're finding the methane. Now, how that energy is getting down into the saltwater section is not quite clear. The haloclines are very important because they're not only a change in salt, but they're a change in density. So if you have a particle that's sinking down through the water, a heavier particle, it can reach that density gradient at the halocline, and it can actually stop and float there on the halocline. So there's a lot of organic matter, particulate matter, that tends to accumulate at haloclines, and we have a lot of organisms that seem to be feeding off or grazing off this crop of food right at the density gradient in haloclines. that's another thing we need to look at, okay? So there's a lot of questions. There's many, many questions we've got. Question, yes?
Going back to that 200-mile long cave system, how
Yes.
did you guys explore it, or how did they explore it, and how long did it take?
So its exploration has been going on probably for 20 years. There are well over 100 different entrances to the cave, but many of these are out in the jungle. They're not in civilization. So what the explorers did was they would start at one cave and they would swim out as far as they could go in the jungle until they popped up in another cave entrance, another cenote, come up to the surface, kind of try to find out where they were, get a GPS coordinate, and then they would go back down and swim home. For the next trip out, they would get some horses or mules, they would load on their scuba tanks, they would get some local people with machetes, they'd go and chop a trail through the jungle where there was no road to get out in the middle of the jungle to get to a cenote that probably no one had seen for 1,000 years, and that would be, they'd set up camp there and start exploring out of that cenote and going as far as they could go till they'd find another cenote and set up another point where they could explore to that. So it was a leapfrog type of situation that took years and years and years of very, very hard work, because the cenotes aren't next to roads or anything. They're in the middle of the Mayan jungles. Not the most pleasant place. There are several very, very poisonous plants in there that can do great deals of harm, that are more than just uncomfortable. One of my students had one of the trees drip on him when it was raining, and it literally ate a hole through his skin. So all sorts of nasty stuff. There's all sorts of bugs and horseflies and mosquitoes and everything that makes your life more difficult. So yeah, it's a great place. But there have been very dedicated divers who've dedicated their life and their career to exploring these caves.
How do you confirm that you discovered a new species? You take the sample and you go to the surface with it, and then...
Okay, how do we find out if we've discovered a new species? Well, first of all, they've been doing this for a while, so I kind of know what to expect. And if I see something, I say, wow, I've never seen anything like that before, that's a very good sign. And a lot of times I try to bring experts along on trips with me that can help identify in the field. If not, when we get out from a dive, the first thing we do is we take a digital photograph under the microscope. So we get a color photograph, high quality photograph, and in this day and age, just send an email on the internet with an attachment, send the photograph off to whoever the expert on that group of animals are, and oftentimes within a few hours, I've got an answer back that says, hey, yeah, great, send it to me, okay? And once I send it to them, they work on it and come up with a description, and I collaborate with them on publishing a paper where I describe the cave, the environment, the water chemistry, what other animals are there, what the behavior of the animal was, etc., in addition to describing the animal itself morphologically, what it looks like, as well as what we're doing a lot of DNA sequencing these days, where we're able to look at the genes, the DNA, and actually compare it with everything else that's known. So oftentimes I know instantaneously when I see something in the water. I've been doing it long enough, or if not, sometimes within a few hours after coming up to the surface and sending off photographs, or if not, by the time the experts have looked at it, have dissected it, and have done DNA sequencing, we get answers back then. And once we've got it, a new species, we need to find a name for it. So we're always looking for creative names or even people we can name things after. So much better to have an animal named after you, a cave animal, than a star, which is a scam type of process anyway. This is for real. So buy a cave animal to have named after you. Here you go. Yes?
What is your mix on that 200 or 165 meter dive? What are you diving in? Is it argon or helium?
So our deeper dives, we're using trimix, so a mixture of helium, nitrogen, and oxygen. It's very important to reduce the amount of nitrogen in our breathing mixtures for doing any deep dives. Also, because we're going down deep, there's great pressure, meaning that the partial pressure of oxygen becomes very high the deeper we go. So oftentimes we're working with very low percentages of oxygen in our breathing mixture. There's gas mixtures that we couldn't breathe on the surface, that we'd drop over and pass out if we tried to breathe them on the surface, but we need them that low to be able to breathe them safely at depth. So my diving, I fortunately have a lot of friends who are the world expert divers, the best in the world. And I have these people come with me because I'm busy doing the science. I'm very occupied. I need someone who I can trust. I need someone who's got my back, okay? And so I get the best people in the world. The photographs, okay? I'm busy with the science. I don't have time to take photographs. So I get the best underwater photographers in the world to come with me and dive with me. Everybody has their job on a team. My job is the science. Somebody else is the safety. Somebody else is the photography. Somebody else is mapping the caves. Somebody else is pushing and exploring new parts of the cave. We have a team made up of experts. Everybody has their job. Everybody has their mission. And everybody does exactly what they're doing the best at. Okay, so it isn't cheap. The rebreathers are at least $10,000 plus the training, so it's a big investment. But not only just to get trained in it, you need to be experienced at using it. You can't just get a driver's license and then you're an expert driver. You can't get a diving certificate and you're an expert diver. So you need a lot of training, a lot of time, a lot of experience. And that's all money. That's all people who are very special people. And I've been very fortunate to be able to get very special people to come out with me. But if you look at other areas of research, oceanic research, look at the deep sea hydrothermal vents. You need a big, huge research ship. You need a submarine to go down. You can only put a couple people in the submarine at any given time. It costs enormous amounts of money. Here we can fly to the area we're going to go to. We can go down and we can stay in a little motel or hotel somewhere, and we can do our dives. In comparison to the amount of money that ocean research takes, our research is damn cheap. So you're getting a lot of value for your dollar. And I think we're into an environment that is as cool as some of the things they're finding in the deep sea, we're finding in the caves right under our feet. So it's right down there. All we have to do is find a way under our feet. And there we are. So it's right here. We need money, but we don't need huge amounts of money like we would if we were studying deep sea organisms. You get value for money with the work on the caves. Yes?
So there's a lot of talk about species and things like that. I was wondering if you could comment a little bit on the isolation of these caves and evolutionary pathways for these organisms, and if there's similarities in those evolutionary pathways.
Okay, good question. Excellent. So, oftentimes, when I first started this work, I thought, well, the animals we're finding in the caves are going to be very similar to the animals in the ocean around the caves. Not true. Bam. Shot down one hypothesis right off the bat. So you have very different animals in the caves, unique forms of life found nowhere else. And second is that if you go around the world, you have the same types of animals found in the Caribbean as are found on the opposite side of the Atlantic in the Canary Islands, as are found in the Indian Ocean in Western Australia or Christmas Island. So how did these animals get distributed around the planet, but only in saltwater caves and nowhere else? So it's the habitat that's the key. Now, in Bermuda, for example, we had animals that we found in 2 caves, and their closest relatives was found on the opposite side of the Atlantic in the Canary Islands. And it turns out that the species were more closely related to the cave and the animal from the Canary Islands than they were to each other in caves a quarter-mile apart from one another. So distance, proximity means nothing. All sorts of strange things we're trying to figure out. So the hypothesis is, the working hypothesis now, is that these animals were distributed by plate tectonics and seafloor spreading. The Atlantic Ocean began forming about 150 million years ago and began gradually, slowly unzipping, spreading further and further apart. So the working hypothesis is that these animals were originally living in the ocean surrounding the megacontinent when all the continents were together in one single landmass. And as this continent fragmented, they drifted along with the continents they were closest to. So they're really ancient forms, and we're hoping that our current studies looking into DNA sequencing of organisms from different parts of the planet will help to shed light on this. So how similar, how different are animals from different areas of the Earth? So we hope to get some answers soon on that. There's one of my graduate students is currently in Florida doing work at a lab there, trying to figure this part out. So we got a lot of ways to go. We're slowly working toward an answer. What happens, well, we don't know. It's always cool. That's the cool thing about this is we've got new discoveries to make all the time. We're being surprised all the time, and we've got all sorts of evolutionary questions that are popping up, and we believe that the caves are a perfect area for testing various theories of evolution of life on Earth. So it goes back hundreds of millions of years. We have animals that are living in these caves and survived when the dinosaurs went extinct. These are animals that were around before the dinosaurs came around, and they lived after the dinosaurs. Man, really gets me. We've got some cool stuff there, and I want to get answers. I've got so many questions and so many answers I have yet to find out. I could work ages on this.
That's great. So I'm going to end it there. Tom's staying with me, so he is going to be here until I leave. So if you want to stay and talk with Tom, he is here. We'll probably take him up to one of the breweries afterwards. But thank you so much. Let's give Tom a round of applause. So how many of you guys are interested in astrobiology here? Is anybody interested in that? So if you're going to study astrobiology, why wouldn't you be talking to this person? Why wouldn't you be diving in these caves? You're going to have to learn about these extreme environments. And I guarantee you, if you're going to find life in some of these moons that we have in our solar system, you probably want to have a conversation with Tom tonight and find out how I can get into be one of his graduate students at some point. So please go talk with him. So it's coming out. But we have the tech to do it here. So think about the wearable tech, the sequencing tech, the microbiome that supports all these apex predators. We can start training these tools here. And actually, we should be talking about launching our own expedition to some of these caves to go dive in. So let's start that conversation. Okay. Thank you, Tom. Thank you guys for coming.